Okay, I'll go through the answers to this. So in the first one, oil extracted from the seeds of the crop false flax can be used as fuel. An investigation was carried out into the effect of a plant growth regulator, paclobutrazole, PBZ, on the photosynthetic pigment content of the leaves and the oil yield from the seeds of false flax. And the results are shown in the table. So let's just think what we're looking at here. So it's obviously a photosynthesis question, and we're looking at pigments. So what do we know about pigments? Remember, they absorb light energy. There's three types: chlorophyll A, chlorophyll B, carotenoids. And it's saying that when this plant makes seeds, you can take oil out of it, which you use as fuel. And what they've done is they've added this drug, basically a plant growth regulator, PBZ, and they've looked to see if that's affected the content of the pigments, and if that's then affected how much oil they get from the seeds. So if we look at the table, and I'm going to use a pen here for the first time, the school's got us pens, so if strange things happen, then excuse me. The first strange thing is when I start writing, I always get this wee, tries to make a box or something. So we've got treatment, untreated, and then the photosynthetic pigment content, chlorophyll A and chlorophyll B, you can see is about the same, but for the carotenoid, you can see there's a bit of a difference. When it's treated with PBZ, it goes up to 3.98, and the average oil yield is higher if they're treated. So let's look at the next bit. Absorption spectra for pigments from the treated and untreated plants were produced and are shown in the graph. So y-axis, we've got increasing absorption of light. And remember, the more light being absorbed, then the more photosynthesis should be happening. And the solid line is your pigments from untreated plants, and then the dashed line is pigments from treated plants. And we've got the wavelengths down the bottom. So you can see that it's the same for both of them, except between this point and this point, so that might be significant. So let's see. Use values from the graph to describe the difference in absorption spectra of the pigments from treated and untreated plants. So it is just looking at this difference. So what you can say is, let me just check exactly the marking scheme. So treated plants have got higher absorption, and then you need to say when. So they've got higher absorption between 450 and 600 nanometers. Okay, so that's your answer for the first one. Treated plants have higher or greater absorption between 450 and 600 nanometers. Then the next one, use evidence from the table to explain the difference in the absorption spectra. So it's giving you these two questions here. The first one, it's saying use values from the graph, so that kind of directs you as to what you should have, what you should put. Here it's directing you back to the table. So use evidence from the table to explain the difference. So why have you got this higher value between these wavelengths of light? So if you go back to the table, if we compare them, the treated ones, the only difference is they have got this higher carotenoid content. Chlorophyll A and chlorophyll B is the same, so the difference you would presume is because there is a higher content of carotenoids, which means you're going to have higher absorption. So that is the answer. So for that one, part 2, you should have treated plants have a higher carotenoid content. Treated plants have a higher carotenoid content. The next one, part 3 is a knowledge question. So state one use that plants make of the light energy absorbed by pigments during photosynthesis. There's a few things you could say here. You could say they generate ATP. You could say they produce hydrogen. You could say they use it for photolysis. You could say they use it to split water. You could say they use it to excite electrons. You could say that they use the energy to pump hydrogen ions across the membrane. Then the next one, suggest why seeds from the plants treated with PBZ yield more oil. So let's just go back and look and what it says about oil. So oil is extracted from the seeds, it's telling us here. See, this is this weird thing that happens. Oil extracted from the seeds. So the seeds are produced during photosynthesis. So if they're producing more oil, then they must be carrying out more photosynthesis. So that's your first part. I'm going to try and attempt to write here. It's not bad. More photosynthesis, so that's for the first mark, and then the second mark is for saying that that produces more energy for growth, or you could say more energy for oil production or more energy for seed production. But that's your 2 marks for that one. Okay, the next one, number 2, the diagram shows crosses in a breeding programme involving different breeds of sheep. So we've got Scottish blackface female times Border Leicester male, and that gives greyface female, and they are then bred with Suffolk male, and then you get your commercial lambs. So part A, suggest a reason why breeding programmes such as this include crossbreeding. So it's basically what is the benefit of crossbreeding. So it's a knowledge question, and that is that the crossbreeds may have improved characteristics. Okay, so to produce better characteristics or better genes or better lambs, better traits within the lambs. Or you could say to get the best characteristics from both, or to introduce new desirable characteristics. Part B, explain why greyface sheep are produced by crossbreeding Scottish blackfaces with Border Leicesters instead of breeding F1 greyface sheep together. Okay, so here's your greyface sheep, and it says, why are they produced by crossbreeding the Scottish blackfaces with Border Leicesters instead of breeding F1 greyface sheep together? So the answer is, if you were only to breed the grey face together, you would only have their genes. So if you breed them with the Border Leicester male, then here's the answer. You get increased variation, or you get increased variety. Or you could also say not all of the offspring will have the desired characteristics that you want. And then I missed out that one because that was taken out of the course. Part 2, explain the importance of selecting a Suffolk male homozygous for the desired dominant characteristic. So let's imagine the characteristic was denoted by H. This is a bit of Nat 5 revision. So this one here is homozygous, and this one here is hetero. So why would you want it to be homozygous? Well, if it's the dominant characteristic that is desirable, then if it's homozygous, then it must pass this on to its offspring. Whereas if it was heterozygous, then there's a chance it could pass on this, which you don't want. So the textbook answer is so that all, or there's more of the offspring which have the desired characteristic, or have the dominant gene, or have the dominant allele. Okay, so more offspring would have the desired characteristic. And then the last one, so apples and plums are grown in North America as food crops. Brown stink bugs feed on apples and plums, reducing fruit yield. Insecticides are often sprayed onto fruit crops to help control these pests. State how the use of insecticides can be harmful to the environment. So one word answer here would be enough to give you the mark. Just thought I'd forgotten how to spell bioaccumulation there. That looks like a 7-year-old writing, but anyway, bioaccumulation, so that's, yeah, you get a mark for that. Or you could just say it could be toxic to other species as well, you could say. Or you could say it persists in the environment, or you could say it could eventually result in resistant populations of pests. Then part B, so this is from the crop protection part, so it's at 3.3, I think. Wheel bugs are a species of insect native to North America that prey on many different insects, including brown stink bugs. So remember, the brown stink bugs, they feed on the apples, which reduce in fruit yield, so we don't like them, including brown stink bugs, but the wheel bugs feed on them. Wheel bugs are used along with insecticides to reduce the number of brown stink bugs. Explain why this method of control would require the use of less insecticide. So you've got to kind of have this in two parts. So the first bit is that the wheel bugs, so you're going to say the wheel bugs eat WBS means wheel bugs. The wheel bugs eat the stink bugs, SBS, and then you need to say so. That means there's fewer to be killed by the insecticide. Okay, so wheel bugs will prey upon the stink bugs, so that means there's fewer bugs which need to be killed with the insecticides, so you would need to use less of it. And then number 2, name the method of control that involves using insecticides and wheel bugs, so that's called integrated pest management. Integrated pest management when you use more than one type of control. Then the last one, a tick is a small invertebrate. Some ticks carry the bacteria B. Burgdorferi in their gut after biting infected animals. If these ticks bite humans to feed on blood, the bacteria may enter the human bloodstream and cause Lyme disease. Use the information given to suggest why ticks can be described as vectors and then as parasites. So use the information given. So we're just looking up here. So why would they be vectors? And there it is there. Okay, so that's the first part. So that's the first part of the answer. So ticks carry the bacteria and they carry it to humans. So they transfer the bacteria or they transfer the parasite to humans or other animals. And in the second one, parasites, why are they described as parasites? And so you're thinking about what your definition of parasite is, and that is that they take blood from the humans, or they take food, or they take energy from the humans. So it's this bit here that they feed on blood. So vectors, the answer was transfer bacteria to humans, and then parasite is because they take blood from the humans. And also just make sure you say for the parasite bit that this harms the humans as well. So they take energy from humans who are harmed in the process. Okay, and then part B, the table shows the average global temperatures between 1981 and 2010 and the number of cases of Lyme disease in the UK between 1999 and 2009. So we've got here table 1, average global temperature for windows of time, and then we've got exact years here where we've got the number of cases of Lyme disease in the UK. So first one, calculate the percentage increase in cases of Lyme disease in the UK between 1999 and 2009. So we are dealing, that's really annoying. If anyone can tell me how to fix that, then I'll give you £1, £1.50. So we're dealing between 1999 and 2009. So percentage change, remember, is change over original times 100. So the change is 670. The original is 200. So 670 over 200 times 100, which will give you an answer of 335%. Part 2, it was concluded from the information in Table 1 and Table 2 that the increase in the number of cases of Lyme disease in the UK was caused by an increase in temperature. Suggests why this conclusion might not be valid. So they're really asking you to use both tables here. So you can see it has the number of cases have gone up, and you can see that the global temperature has gone up, but we're not really comparing like with like here. So that would be one potential answer. So the temperature measured is global, whereas Lyme disease in the UK, that would be 1 mark. Or you could say the temperature and Lyme disease are over different times of the year. You could potentially say that. So that would be another one. Or you could just say, well, maybe it's not temperature. It could be another factor which has affected it, which hasn't been measured. Okay, another variable such as whatever could increase the number of, could cause an increase in the number of ticks. Could be an increased number of cattle or something. But the main thing is they've compared global temperature and they've measured in the UK. And then finally, 2 methods used to reduce the number of cases of Lyme disease in humans were suggested. So number 1, use pesticide to kill ticks, and 2, treat infected animals with antibiotics. Explain how each method could reduce the number of cases of Lyme disease in humans. So method 1, if you use pesticide to kill the ticks, then you've got fewer ticks, and then you need to have the second bit. So you get fewer ticks, which means fewer ticks to carry the bacteria, okay, which caused the Lyme disease. You have to link to that. So fewer ticks to carry the bacteria or to spread it. Or you could say the ticks have been killed so they can't spread the bacteria. Then the second one, treat infected animals with antibiotics. So that would mean you would have less bacteria in the animals, so you have fewer infected ticks to transmit to humans. Okay, or less bacteria, so that means there's less chance of the disease being passed on to humans. Or you could say when the tick bites the animal, it will not pick up the bacteria, which means it can't pass it on to humans. And that is them all.